the fifth week of the stem cell training program, and we are continue to observe the new participant registration still in the middle of the program. And for those who joined recently, I'm Gitai Kim, Director of the UCLA Human Stem Cell and Genome Engineering Center. And our center is just focusing on the both collaborative research and the core service in iPSC programming and the genome engineering. Some of the institution I access through the core-to-core partnership, so therefore you will be familiar with our center. And this program is supported by the California Institute for the Regenerative Medicine in collaboration with Thermo Fisher Scientific. And a number of the staff members is involved, but the main leader is Dr. Omar Farah and Dr. Thomas Forbes. And as you see here. And as a reminder, and the participants who attended at least 6 out of 7 lectures will receive the certificate of the training completion. And even if you have already missed 2 lectures by joining late, but you can still qualify by attending the mixed session next year. We will provide the next year too. So please continue to attend the lecture. And for the attendance tracking, and then please log in to Zoom using your full name, first name and the last name. And if you are logging in with your partial name or different name, please write your full name and the email address in the chat box to make sure we are tracking your attendance. And if you are attending without registration, please report to the chat box. And I also attached a number of the useful link and those one, and then you can register. And you are welcome during the lecture, and you can anytime just speak up, turn on your mic, and then just directly ask the question, or just post it in chat box, and then other staff members will continue to answer your questions. And then today we will discuss about lecture 5, very hot topic, gene editing in human pluripotent stem cell, focusing on the genome engineering strategy and the practical application in stem cell system. Now I would like to hand it over to Tom and Omar.
Thanks, Gitai. Let me just share my screen here. Share. Can everybody see that okay?
Yes. Yep, perfect.
Good. And everyone can hear me okay?
Excellent.
Cool. All right, let's get cracking in. Well, hope you all had a nice weekend. Thanks, Gitai, for setting the stage for today. I'm going to get going in the interest of time. We've got, I think, a really interesting topic to cover today and quite a lot of ground to get through. But as you see on the slide here, today we're going to be talking about gene editing. We're going to talk just very briefly initially, a couple of slides on optimized workflows. Again, this is always, I think, a nice opportunity to review a bit of the content that we've covered so far. But I think it also serves as a nice segue to sort of set the stage for gene editing. And we're going to spend the bulk of our time today really talking about 2 different things. And the first is, as you see here on the slide, in regard to some really interesting technology surrounding designer engineered nucleases, specifically CRISPR and TALENs, which you've likely heard of, at least one of. CRISPR especially is pretty famous, I would say. And then, as we often like to do, I thought it'd be really interesting at the end to put some of this content into a case study. So I picked one from the archives on our end, and I'll be talking about gene editing in the context of cardiac disease modeling. This is work that was done in 2D, but I think it also sets the stage a bit for next week's lecture, which is going to be a lot of 3D content given by Omar, and we have a special guest lecturer as well that'll go after Omar, Dr. Glauco Souzer. He's from Grenier Bio 1, I believe is the company. And they're both going to be talking to us about mesodermal differentiation, which of course includes the heart. And then Dr. Souza will go over some really interesting principles surrounding 3D culture and I believe new approach methodologies, or NAMs, which is another really hot topic at the moment. As I've done before, I think this is a nice slide to set the stage. We always try to tell a comprehensive story, right? We start, as we often do, with a particular cell model in the left here. Again, we isolate somatic cells from donors, for instance. We reprogram, as Omar talked about in our reprogramming lecture, to become those iPSCs, those induced pluripotent stem cells. And then in a lot of modern day workflows anyway, we engineer those cells, and we do that to program specific target nucleases to sites in the genome. We're going to talk about some of that today. When we're doing things like genome editing or reprogramming, right, it's especially important to characterize your stem cells that you're working with. So I'll talk a little bit about that as well today. Obviously, this is something that we do to give us good peace of mind as far as our experiments are going and progressing. But I think it's also an important thing to do, the characterization, to take stock of where you are, right, and confirm, especially with your cells, things like identity, genomic stability, pluripotency, and other important characteristics of stem cells that we've talked about so far. Before, nowadays, as you see over here on the right, we often take those edited cells and differentiate them into physiologically mature, often germline specific cell types that better allow us to mimic relevant disease states. And so along those lines, as you heard me talk about before, good cell models, right, they have the capacity to, as I've said, predict outputs that translate to organisms, right? In other words, they're physiologically relevant with regards to human biology. And as we've talked about, these different outputs come in loads of different flavors, right? We've got gene expression, protein function, cell phenotype, cell health, the list goes on and on, right? But basically, it's from all these different inputs across the top here that we're able to then generate data or different outputs, right, which we then decipher and ultimately translate back to the organism. Again, in our case, relating, and I would say making sense of all these results, hopefully, back to the human state. Now, as I said, as Omar talked about before, with the advent of iPSC technology, there's been some really, really interesting advances that have allowed us to better these models and correlate them even more to human disease, right? Even honing in on the contributions of, say, a single gene with respect to a particular disease, right? And as I alluded to at the end of this talk, we're going to do that in the context of a heart disorder, and that heart disorder is called dilated cardiomyopathy. And so once we have those cells on our model for in vitro modeling, right, the next stop in the workflow is reprogramming, which we covered again in the second lecture. And again, that's that process of reverting, if you will, a somatic or adult cell almost back in time to a pluripotent state so that it behaves like a stem cell. And we do that by forcing the expression of 4 different transcription factors, right? OCT4, SOX2, KLF4, and c-MYC. And as we talked about, there's loads of different ways to deliver these different factors into the cell to start that process of reprogramming. And we're quite spoiled nowadays. Lots of smart people out there. There's a lot of different techniques at our disposal, some of which we've listed here. Again, from a bird's eye view, though, most reprogramming applications deliver these transcription factors through either integrative or non-integrative methods. I'd say more traditional technologies. So think things like lentiviral delivery, for instance. What those do is they integrate into the genome of target cells versus, say, non-integrating methods, as you see here in the green boxes, right, which generate iPSCs that don't contain detectable vectors or transgene, the most efficient of which are sendivira particles and episomal DNA vectors, which I've circled here in red, both of which we offer at Thermo, and both of which support the reprogramming of a wide range of cells. So switching gears here slightly within the workflow, right, and continuing on with our story, right, genome editing is really the meat and potatoes of what we're aiming to cover today. So again, a really just, I think, nice, simple analogy is think of editing in a Word document, right? We're adding, removing, we're replacing and changing words. So genome editing is really an approach in which the genomic sequence is directly changed by adding, by replacing, or by removing DNA bases. And you heard me maybe say this in the introductory lecture, but the genome itself, it's important to consider, is relatively resistant to change, right? DNA in the body is responsible for loads of different things, right? Encoding all the necessary functions, if you will, within a cell. But it's also really, really important in determining differences, right? Individual differences between people, between organs, between different things going on in our body. And so if DNA was very easily altered, a lot of essential cell functions could very much be disrupted, maybe in undesirable ways, right? And so what we've done is to protect against those inadvertent changes. Cells have almost developed these sort of inherent mechanisms, if you will, to proofread and repair their genetic code, which is something that we actually tap into when we're performing gene edits. And when you use gene editing, right, to produce or introduce a desired phenotypic trait into a cell or an organism, right, you create much, much better cellular models. These cellular models can be used in a whole bunch of different applications, right? I've listed a few here, but functional genomics, drug discovery, cancer research, epigenetics, and so on and so forth. As with any scientific application, again, there's some really, really smart people out there that have allowed for some pretty hefty advances over the course of time. As you might imagine, genome editing from sort of its early conceptualization through to the advent of technologies like what we'll talk about today, like CRISPR-Cas9, has quite a rich history, as you can see here, evolving all the way from sort of a very, very basic setting all the way to various applications nowadays in medicine, agriculture, and beyond, right? So this is a fairly general timeline, I would say, that I pinched from Nature. It's a couple of years old now, as you see. I think it's from 2016. But you get the idea of sort of that evolution of some of these genome editing methods, all the way from, say, yeast models to things like zinc finger nucleases, and again, more recently, things like designer engineered nucleases like CRISPR and TALENs, for instance, which we're going to be talking about shortly. And there's a lot of different angles that we can take nowadays to genetically engineer those cells, right? Techniques like gene repair, overexpression, the addition or the deletion of certain genes, all of which, again, we tap into and really try to take advantage of to build better workflows, as you see across the bottom here, and then further investigate underlying disease mechanisms, again, to bring it back all selfishly again to help us understand human biology. So this slide, from a sort of a high level, if you will, highlights some of the current strategies underlying genome editing, whereby we're doing things like inserting defective genes to generate relevant models, or maybe we're inhibiting certain genes to render disease-causing pathways inactive. We might be inserting drug-sensitive markers that allow us to test, say, the safety or the efficacy of drugs. The list goes on and on here, right? But gene editing is, as we've talked about, a really, really cool and I think rapidly evolving space. And some of the capabilities nowadays are just incredible. So again, I think as good a starting point as any to jump into a bit of detail here is with these designer engineered nucleases, right? And again, these include strategies like CRISPR and TALENs, which can be designed to target nucleases to specific sites in the genome and then essentially create double-stranded breaks, right? Whereby that, as you see here in this top red arrow here, following those double-stranded breaks, the natural repair mechanisms of the cell actually help heal those breaks. This is done by typically things like homologous recombination or non-homologous end joining. And again, we'll go into a little bit more detail on those in just a moment. And so, as I alluded to, I'd say CRISPR technology and TALENs are things that essentially reprogram target nucleases to very specific sites in the genome, and again, allow us to create those double-stranded breaks at specific locations. And we're going to start today with, I'd say, the most popular and arguably the most famous of these engineered nuclease systems, and that's CRISPR-Cas9, right? So CRISPR stands for clustered, regularly interspaced palindromic repeats. Total mouthful, absolutely no need to remember that. But this is, from a high level, an RNA-guided DNA nuclease system that, as I mentioned before, was originally identified as part of the bacterial immune response pathway. Specifically, though, in something called Staphylococcus pyogenes, as we see here on the right, where we know that they're part of their adaptive immune system that helps protect that organism against invading DNA. What scientists did, though, as they often done, is sort of pinch that notion or that idea and sort of further that and gained a better understanding of this molecular pathway, and then co-opted it for use in eukaryotic engineering, right? And so ultimately what we need for CRISPR-Cas9 is an RNA-guided DNA nuclease system, right? So that means bacterial enzymes that cleave double-stranded DNA. And that nuclease or enzymatic activity is conducted by an enzyme called Cas9. You could think of Cas9 on the left here, almost like a pair of molecular scissors, right? It can cut the two strands of DNA at a very specific location in the genome. When you do that, it enables bits of DNA to be added or removed. We also have a piece of RNA called the guide RNA, and this is designed to essentially find and bind to a specific sequence in the DNA. And the guide RNA has RNA bases that, as you might imagine, are complementary to those of the target DNA sequence. That means, at least in theory, right, that the guide RNA will only bind to the target sequence and no other regions of the genome. And so this guide RNA, or gRNA, usually consists of a very, very small piece of pre-designed RNA sequence. It's usually about 20 bases long or so. That's often located within a longer RNA scaffold. And the scaffold part binds to the DNA and guides or recruits Cas9 to the right part of the genome based on that pre-designed sequence, basically making sure that the Cas9 enzyme cuts across both strands of the DNA at exactly the right spot. Now, in the case of Cas9, the complex actually steers and integrates a really small piece of foreign DNA, something called a PAM, PAM, right, or protospacer adjacent motif, another mouthful, into the CRISPR loci in its chromosome. And for S. pyogenes, this PAM motif is always an NGG sequence. So the PAM sort of acts as a binding signal, if you will, for the Cas nucleus, allowing it then to distinguish between foreign and host DNA, and hopefully ensuring that the CRISPR locus itself isn't actually targeted and destroyed. So the big take home here with CRISPR-Cas9, it's a novel gene editing system. The system really consists of two parts, the Cas9 enzyme and the guide RNA. And this allows us to modify, edit, delete, or even correct specific areas of our DNA, again, by capitalizing on an endogenous repair mechanism. What happens next, though, sort of taking it and diving one level deeper, I think is really interesting. Because as I said, right, the cell recognizes that the DNA is damaged, and so it tries to repair it. And so what we have here is a cartoon representation of what's happening at the molecular level. In red at the top here, we've got a double-stranded piece of DNA, and this gray circle here represents an engineered nuclease that's going to come in and cleave that DNA, thereby causing a double-stranded break. When that break occurs, as I said, the cell's going to attempt to repair it, right? When it does, there's generally two main pathways or natural repair mechanisms that are involved in this sort of healing process, if you will. The first is non-homologous end joining, or NHEJ, shown here on the left. And this is the primary pathway for the repair of DNA double-stranded breaks throughout the cell cycle. And we refer to it as non-homologous because the end breaks are directly ligated without the need for a homologous template. And these are generally what's used to make knockout models or perform genomic screens, right? And they also occur quite quickly, and they have a high capacity for repair because, again, there's no requirement for a repair template, and therefore no extensive DNA synthesis. The second healing process on the right here is called homology-directed repair, or the HDR pathway. And homologous recombination is a bit more precise because it does require that specific template for repair. And so by providing the cell with this synthetic template containing a sequence of interest, for example, a disease-specific mutation, the researcher can introduce this sequence into the genome. And this is what's done to perform gene insertions or general knock-in editing. Now, for HDR to occur, we require the presence of a donor molecule, right? One that contains the new DNA that needs to be added to the genome. And as you can see in this figure here in the right, those left and right homology arms of the sequence flank that break site, right, which allows the donor template to be incorporated into the genome right at that specific spot. And generally speaking, there's three main materials required here. The first is a specific and efficient nuclease system. The second is a repair template with the desired change. And the third is a method to deliver them into the cells. And so it's really the collection of these materials together that causes this edit to occur at the molecular level. And then last but not least here, I want to point out there's really four main types of donor templates, if you will, used by scientists today. And I've listed at the top here those in order of increasing size. The first is single-stranded DNA. And this is generally used for quite small insertions like SNP changes or amino acid substitutions, typically on the order of less than about 200 nucleotides. Second is double-stranded DNA. And this can be used for larger insertions like fusions or reporter genes, typically on the order of about 200 to 5,000 base pairs. Third, we have plasmid DNA. And this is used for very large insertions, typically up to about 10 kb. And then lastly, viral vectors, specifically things like AAV or adeno-associated virus, which are generally reserved for really big insertions in cells that are difficult to transfect. We generally use the first two at Thermo, right? Single-stranded and double-stranded templates. Now, one other thing I think that's important to call out here is that with CRISPR-Cas9 systems being so widely used, there's some, obviously, and rightly so, emerging concerns about off-target effects when we're using this specific approach. And these off-target events are something that occur when unintended edits happen, right? And this potentially, as you might imagine, adds another variable or unknown to a particular experiment. So I've highlighted here a few papers along those lines if you're interested in some further reading, and really just want to emphasize again that it's important to monitor and minimize those off-target effects, especially when you're considering therapeutic applications, right? Because this allows us to reduce the risks associated with those undesired edits in the experiment. But how on earth do we do that? So this slide is something we pulled together that demonstrates a couple of different strategies that are available to reduce those off-target effects. For example, on the left here, we've got an RNP delivery format. Those RNPs, or ribonucleoprotein complexes, are essentially a protein-guided RNA complex that provides a very initial high level of CRISPR-Cas9, followed by rapid decay. And this sort of transient approach, if you will, helps minimize the risk of off-target generation. Another strategy that we employ is something called a high-scoring guide RNA to minimize off-target effects. This is actually a computer simulation that we offer at Thermo Fisher in the form of something called our True Design Genome Editor, which basically enables us to predict potential off-target loci by providing a sort of guide algorithm, RNA algorithm score, right, that predicts things like efficiency, specificity, and proximity to the edit site. And then lastly, we often employ the use of something called high-fidelity Cas9, and this is essentially a version of that Cas9 nuclease that's engineered for high specificity or reduced off-target effects. And again, I don't want to go too far into the weeds here, but I think this is super interesting. That high-fidelity Cas9 protein is, as I said or alluded to, a variant of the Cas9 nuclease, which, as a reminder, are those molecular scissors, right? And they help improve specificity, in this case, by the incorporation of additional point mutations. And what those point mutations do is they destabilize interactions between the Cas9 and the target DNA. And that sort of tailored destabilization, if you will, makes the high-fidelity Cas9 much less likely to bind to non-complementary or off-target sequences, therefore resulting in improved specificity. If we compare the two, right, performance between, say, the wild-type Cas9 and the high-fidelity Cas9, it's really a balance. We're always trying to strike a balance here between on-target editing efficiency and really the ability to reduce off-target effects. So, for example, with the wild-type Cas9, we do see typically superior on-target editing efficiency, but it comes with a trade-off of more off-target events. In contrast with the high-fidelity Cas9, we expect improved specificity, right, and therefore the ability to reduce off-target events. But the kind of trade-off here is a decrease in on-target editing efficiency. All just food for thought. And then the last thing I wanted to touch on here with regards to these Cas9 nucleases is that they can, as you see here, take many forms, right? In the early days of CRISPR, scientists primarily used Cas9 expressed from a plasmid, but I would say that quite quickly fell out of favor, mostly for efficiency reasons. Nowadays, though, protein and mRNA formats of Cas9 exist, right? And they offer, as you might imagine, DNA-free, transient expression without a lot of those residual effects that you'd see with, say, a plasmid. And our version of the Cas9 protein is something called TrueCut Cas9, which is quite popular for both knock-in and knock-out experiments. Okay, if we go back to this slide again, this time around, I want to switch gears again and focus on the other gene editing system that we'll be talking about, TALENs. And TALEN stands for transcription activator-like effector nucleases, as we see on the right here. And these are based on effector proteins that are secreted by certain types of bacteria, right? Bacteria that we know actually cause major diseases in crops. They're an absolute nuisance for farmers. And so they use these pathogenic proteins to basically rewire transcription of plants that get infected, right? And then induce expression of certain genes, albeit in a much simpler manner in plants. So how does this work? Well, as I mentioned, these TALENs are based on effector proteins secreted by certain types of bacteria, specifically something called Xanthomonas bacteria. And so these TAL nucleases or effector proteins consist of repeat sequences, usually about 33 amino acids or so. And these sequences or motifs have variable positions, right? Those variable positions, that's very important, that recognize specific DNA nucleotide sequences, basically allowing for just about unrestricted targeting of any sequence in the genome, right? And what happens is the TAL effector DNA binding domain binds to this nucleus here called focal, right? F-O-L-K-L. When we form them, that allows us to then form a dimer that induces breaks in the DNA, resulting then in a frame shift mutation in subsequent genes that results in their knockout. These talons, though, also quite nicely are now available in an mRNA-based format, making them even easier to get into stem cells. And a big part of why they can be addressed anywhere in the genome, because there's no PAM motif constraint like we saw with CRISPR. Now, talons, I will say, are perhaps a bit more limited to simple mutations, unlike, say, CRISPR, which can introduce, say, multiple gene mutations concurrently, even with a single injection. CRISPR transfections also tend to have a higher efficiency, whereas talons can sometimes result in something called a mosaicism, which is basically where you have a mutant allele that's present in only some of the cells that are transfected. Now, as far as delivery goes, these are some of the tools on the right here that researchers nowadays have at their disposal. As you can see, there's quite a few options to carry out that process of gene transfer, as we can see in this cartoon here. A good deal of what we employ at Thermo relies on either lipid-mediated transfection or electroporation. So if we start on the left here, though, with lipid-mediated transfection, this is a technique in which we, as we talked about in the introductory lecture, essentially take advantage of the positive charge of lipid complexes to bridge with the net negative charge of the cell and DNA, right? The complex then enters the cell via endocytosis, as we see on the bottom left here. And I've not listed them here, but there's obviously a number of factors that can impact this process of lipid transfection, things like cell type, passage number, cell density, cell health as well, even the reagent amount and the ratio to delivered molecules. So all really food for thought when designing and implementing an experimental plan. We also at Thermo rely quite heavily on the process of electroporation, which I've circled down at the bottom here. This is quite a physical process that uses an electric pulse to basically create a temporary pore in the cell membrane. And this is quite an efficient strategy for introducing nucleic acid to the cells, whether, again, for the creation of stable cell lines or just for a transient transfection. But basically, host cells and biomolecules to be transfected are placed in a conductive solution of some sorts. We then apply an electric pulse at a set voltage, right? And what that does is it disturbs the phospholipid bilayer, resulting in the formation of almost this temporary pore, right? A hole in the cell membrane to which DNA is able to pass. These pores, pretty cool, can remain open for quite a few hours. But what that does mean is that the cells need to be handled quite delicately, even after they've been plated. So they are a little bit finicky at times. Electroporation typically also requires or relies on either cuvette-based technology or in some cases more advanced pipetting-based strategies, which I'll show you a little about in a moment. But this is, I think, a really important and really, really incredible technique, especially for difficult to transfect cells. Now, again, we won't go into too much detail here, but just so you have it as a nice point of reference moving forward, I've listed here a couple of pros and cons of some of the different delivery methods, including things like lentiviral delivery and microinjection. So just a nice point of reference for you guys if gene delivery is something that you're interested in taking a deeper dive on. Just quickly, quick shameless plug here at Thermo, we do use our Neon Next electroporation system for physical transfection. It's really easy to use. It's fast, it's efficient system. It allows you to optimize a lot of different parameters surrounding electroporation, again, especially for hard to transfect cells. But the system also comes with quite a couple of hundred, I believe, pre-optimized protocols over about 150 cell lines or so. So it's quite a user-friendly way to electroporate your cells. Now, I think it's also worth a mention here. This is a website that we have at Thermo Fisher. But as I said, is an online tool to search for and order pre-designed guide RNAs. This is a website you can go on and just play around with if you want. It's pretty fun to sort of tinker with. Again, it uses an algorithm to help in the design of these guide RNAs for effective gene knockout. We have synthetic gRNAs available for both mouse and humans as well. And we also offer human designs for lentiviral guide RNAs. Another I think really interesting and fun tool to play around with is our True Design Genome Editor software. This is something that allows you to design and order your CRISPR guide RNAs, the associated DNA donor, and even add other carts to the cart if you so choose, all from this one tool to help you sort of complete an experiment, if you will, and kind of give you, I think, a really intuitive visualization of the genomic sequence to help navigate this sort of design process. including things like designing knockout experiments using donor sequences, again, that can target just about anywhere in the genome, calling out off-target considerations in, say, the population of interest. And one other kind of shameless plug here is that we do actually have a dedicated team of application scientists at Thermo that wholly support the genome editing workflow. This is something that's a little bit peripheral to what Omar and I do, but some of the genome editing stuff from R&D and from our application scientists that they talk about is really, really interesting. They go into all kinds of nitty-gritty that are well above my head. But if you are doing this and you do want support with this, this is something that we can help connect you with a dedicated expert in this space. Just kind of tying that all together here, this is a nice overview slide of the genome editing workflow, kind of at a high level. In this case, the CRISPR-Cas9 workflow, which of course includes the design and build of the workflow itself, right? We're trying to obtain superior gRNAs and donor designs for a specific experiment. We then want to pair those with high-performing enzymes, right, that allow us to maximize editing efficiency. Culture and delivery is really important too, right? We're aiming to deliver editing tools, obviously, to healthy and viable cells. We want to do that with high efficiency. And then finally, detection and validation, right, where the goal is to confirm your editing efficiency, determine your percent modification, and then help to validate the edit itself. And so this is a fairly all-encompassing workflow for design, delivery, and detection, again, allowing you to engineer your cells all from one place. Data-wise, this is taken from a fairly recent ISSCR poster where we demonstrated the high specificity of that TrueCut Hi-Fi Cas9 system that I talked about, in this case in human iPSCs. So a little bit interesting here in this experiment, what we did is we selected gRNA targeting 3 different genes, HBB1 and HBB2, which are hemoglobin beta subunit genes that we know are associated with sickle cell anemia, as well as something called HEC4, which is a well-studied site for off-target effects. What we're looking at here in the data is TEG-seq, a data plot graph in which the TrueCut Hi-Fi Cas9 registered fewer gray dots or observed off-target effects compared to the wild-type TrueCut Cas9 protein. And we also compare this to IDT's Hi-Fi Cas9 here. And so basically what we're seeing is a reduction in off-target events that we observed at the HEC4, representing on the right here about a 55% elimination of off-target events when compared to the wild-type Cas9. So basically this allowed us to demonstrate that TrueCut Hi-Fi Cas9 enabled higher specificity in these human iPSCs. Similarly, this is taken from, I believe, 2021 or 2022 poster from ISSCR. We looked at off-target effects in primary T cells here using the TrueCut Hi-Fi Cas9 protein. What we did in this experiment is selected 21 guide RNAs targeting 4 different therapeutic genes, TRAC-A, CD52, TRAC-B, and PD1. We did sort of a benchmarking study against the wild-type TrueCut Cas9 protein here, and again, IDT's high-fidelity Cas9, using TEG-seq as our analysis tool. What you're seeing in these dot plot graphs is red dots representative of on-target events and gray dots representing off-target events. The dot is plotted based on the ratio of off-target RPM, or read per million, over the on-target RPM, or read per million, right? So the higher the percent ratio, the more likely the off-target events are to occur. You can see in the graph here that compared to the other 2 enzymes, there's significant reduction in off-target events with the TrueCut Hi-Fi Cas9 protein across all the primary target loci here in those primary T cells. We also saw that the ratio of off-target events was relatively lower than the other nucleases, indicating that these off-target events had a lower probability of occurrence. Now, as far as other really, really important workflow considerations in the genome editing space, we've talked about this a little bit before, but media choice is really, really important in the context of gene editing. As we've talked about, there's loads of different media choices, sometimes far too many, admittedly, even at the same company. This is a table that compares some of those different media options, again, where you can see different advantages for particular applications using one media type over another. In the context of gene editing, though, as we see at the bottom here, we pretty much universally recommend StemFlex, which offers a lot of support and recovery for cells undergoing those different stressful cell culture applications. And again, just a little bit of a shameless plug here. That's because of StemFlex's strength and versatility during those challenging applications, where it shines in a number of different areas, as I've listed here. Things like weekend free compatibility, robust recovery, as well as improvements in things like cell survival. A little bit of data just to support that claim of StemFlex performance in the context of gene editing. What we're looking at here at the bottom is recovery after singularization, electroporation, with the Cas9 protein and a guide RNA. What we did in these experiments here is seeded cells at the same density and then allowed them to recover in different media systems, plus or minus something called a Revitacell supplement. Revitacell is something that we offer that helps with the recovery and overall cell viability. So this is an application where we typically expect cells to probably require, I would say, the addition of Revitacell or another ROCK inhibitor type to allow for their survival in this sort of stressed out cells. But what we found was that StemFlex here showed optimal recovery out of the 3 media systems that we tested, even in the absence of that Revitacell supplement. Again, this is just a little bit more data here comparing recovery in different media systems. Really just highlighting again that media is really an important consideration for these challenging workflows. And you can take my word for it, and I'm sure others, if you've not had this experience, that you can certainly save a lot of time and money and frustration by choosing the right culture system for different applications from the get-go. Putting a little bit of that gene editing into action here and demonstrating a bit of utility of this system. What we're looking at here is making stem cells from either healthy donors or patient donors, and then evaluating disease-specific SNP changes, right? The SNP changes being single nucleotide polymorphisms. Those are DNA sequence variations that occur when our nucleotides, right, adenine, thymine, cytosine, or guanine, are altered in the genomic sequence. What we did here is either corrected those SNP changes or introduced mutations into healthy donor cells to reproduce disease phenotypes. So what we're looking at here is Cas9 iPSCs grown in StemFlex medium. We then used our GeneArt kit to support CRISPR editing, make guide RNAs in order to make those SNP conversions at different locations or genes. We then delivered these guides alongside a single-stranded DNA oligo donor system, again using the NEON electroporation system we talked very briefly about. We measured cleavage here using the genomic cleavage detection kit. On the top here, you see a gel representing results from genomic cleavage. We see here double bands appearing as you increase the amount of guide RNA, again indicating a product that's resulting from that genomic cleavage, which we would then quantify. In the bar graph here, we see percentages of insertions or deletions, also known as indels, right, in blue, or HDR, right, our homology-driven recombination in red, for a variety of loci here that we know are associated with different diseases. And I won't go into too much here, but these include central nervous system disorders like epilepsy, schizophrenia, Parkinson's disease, as well as mutations associated with 2 cardiac disorders, dilated cardiomyopathy and cardiac rhythm disorder. Now, along those lines, and I alluded to this a little bit at the beginning, a really important consideration when working with stem cells, and especially in the context of gene editing or stressful interventions, is characterization. And characterization is that process of, let's call it testing and verifying things like the identity and the quality and even the functionality of your stem cells. So this is a pretty busy table here that I pinched from ISSCR that summarizes some recommendations for basic characterization of cells, including the timing of any characterization. As I said, this is from ISSCR, the International Society for Stem Cell Research. They're essentially the governing body that helps ensure rigorous and reproducible stem cell research. They put out a lot of standardized recommendations, including some surrounding characterization. And they also put out quite a lengthy and very interesting document every year for their guidelines, if you will, for stem cell research and clinical translation. Within that document are 4 different factors or important considerations that they've deemed really, really, really important surrounding characterization, and I've highlighted those here in red. The first of which is basic cell line characterization and maintenance. So in this case, we're thinking about things like authenticating cells against a reference sample, say from the original donor, using specific nomenclature, right, or unique identifiers, and also meeting standard hygiene requirements, right? So rigorous testing for mycoplasma, bacteria, fungus, human pathogens, for example. The second key aspect that they call out is pluripotency and undifferentiated state. So as you might imagine here, they're suggesting assessment of marker expression, right? So think things like OCT4, NANOG, TRA160, often by techniques like flow cytometry or immunofluorescence, as well as assessment of differentiation potential, right? And we often do that through techniques like embryo body formation or directed differentiation to the 3 germ layers. The third suggestion here is genomic characterization. Ideally here, researchers would quite routinely evaluate things like genomic integrity, especially in their master and working cell banks, and then also monitor for acquired genetic changes after culture, after cloning, or after things like gene editing. And then lastly, the fourth principle here is reporting standards. And I think this is something that, I hate to say it, often gets swept under the rug, but that's really, really, really an important thing to call out. Researchers should always maintain a very detailed documentation of their experimental paradigm, right? And that includes things like cell source, the species, the tissue of origin, even, I think, the culture methods that we use, right? The media, the substrates, the seeding densities, and so on and so forth. Especially when you're publishing and you want to go and read a journal article or something that looks really cool, I think it's always useful to know how they did it, some of the experimental parameters that they used, right? And so I think it's important to call that out to make sure that this essential information is always included, if you can, for other people. Okay, deep breath. Last but not least here, I know this is a lot of information, but I did want to share some data from a model here looking at dilated cardiomyopathy. Dilated cardiomyopathy, the simplest way to put it is when heart chambers enlarge and they lose their ability to contract. And this is another disease modeling story that we have through a collaboration with our R&D team at Thermo, one of our customers. And I put this in here today because, again, I think it sets the stage a bit for next week when we're going to be talking about mesodermal differentiation. Omar is going to share with you some of the work that we've done in the 3D space with regards to mesodermal differentiation, as well as some others. And so what we have here is a specific heart disease model where you can kind of see in this nice overview slide here that we're going through, again, the different buckets of our stem cell workflow. In this case, we're using a Cas9 expressing iPSC parental line, engineering a mutation in something called troponin T2, specifically something called TNNT2. This is a mutation that we know leads to dysregulated muscle contraction in the heart, and that happens because of alterations in intracellular calcium. This sort of calcium dysregulation is a very essential component of the disorder that we're talking about, this dilated cardiomyopathy. And again, what we see in humans with dilated cardiomyopathy is that it's usually a disease that starts in your heart's main pumping chamber, right? The left ventricle. And over time, it sort of stretches and it thins, so it dilates, right? And what that means is it becomes weak. It can't pump the blood as well as, say, a healthy heart. And so in this model here, after generating this model itself and then performing gene editing, we characterized the engineered iPSCs, and then we differentiated them into cardiomyocytes, and we have a kit for that, our cardiomyocyte differentiation kit. And then we performed several cell-based assays to basically understand, or help us understand anyway, the effect of this mutation on cardiomyocyte or heart cell function. From that cardiovascular disease model workflow and looking a bit more closely at the characterization process, what we did here is, after introducing the mutation in Cas9 iPSCs, the team reconfirmed targeting of that troponin T2 gene by Sanger and next-gen sequencing. And again, without going into too much detail here, essentially examine the quality of those edited iPSCs using a couple of different characterization tools that I would say collectively allowed us to confirm that things like pluripotency and karyotype in the cell lines that we were working with were not affected by genome editing, things like line clonality. After we confirmed those genomic edits, the Cas9 parental control line and the mutant lines were differentiated side by side into cardiomyocytes. You can see here that, similar to the Cas9 parental line, those troponin 2 mutants differentiated normally, right? Normally, this is evident here with both mutant and control lines staining appropriately for a couple of different markers here, troponin 2 in red and actin 1 in green, both of which we know are structural proteins and essential components of the contractile apparatus in cardiomyocytes. So what this showed us is successful differentiation of cardiomyocytes from both the troponin T2 mutant line and the control Cas9 iPSC line. And then lastly here, we examine functionality in our mutant cardiomyocytes by monitoring calcium transients, right? I talked about the importance of calcium transients, but what we see is under normal circumstances, these calcium transients in the cytosol basically control each cycle of contraction and relaxation in that beating cardiomyocyte. As you might imagine, when we mess with that calcium transient, right, we can see things like an apparent delay in calcium decay over time in our mutant versus our control cardiomyocytes. You can see that in the top left graph here, where they use a specific dye, something called fluo4, to visualize cellular calcium in both the mutant, which we see in orange, and our control cardiomyocytes, which we see in blue. Now, we know that this delay in calcium transients is correlated ultimately with a slower beat rate in the mutant cardiomyocytes as compared to wild-type controls. And this is demonstrated here in this top right panel. And then lastly, down at the bottom here, under stressful conditions, think things like extensive exercise, for example, in humans, what cardiomyocytes will do is they'll attempt to compensate initially by increasing factors like beat rate, basically as a means to try and support increased blood flow. And so along those lines, what we did at the bottom here is used cardiomyocytes in culture and stimulated them with a drug called isoproterenol, right? This is a beta-adrenergic drug that we know accelerates the beat rate of these cardiomyocyte cells. And we used a control called DMSO, which does not induce this effect. This is something that cardiac researchers use quite often to mimic cardiac stress in vitro. And again, we can see the results of this test, the stress test, if you will, down at the bottom here, where we saw spontaneous contraction in the mutants was rapidly compromised after this isoproterenol administration. So again, just to summarize the case study here, we used a Cas9 expressing iPSC line to generate a troponin 2 mutation. We then confirmed that these edited iPSCs maintain pluripotency and a normal karyotype. We then differentiated those edited iPSCs into cardiomyocytes, discovered that this specific mutation, right, affects spontaneous contraction rates, often leading to, in this case, challenge-induced cardiac arrest, and mimicking many of the pathophysiological hallmarks of disorders like dilated cardiomyopathy. Just to wrap up today's lecture, again, I want to say I think we're very spoiled nowadays in the number and the extent of the technologies that we have at our hands. Everything surrounding things like the generation of iPSCs from our somatic cells, which, you know, in many ways allow us to create these basically limitless pools of iPSCs that do retain some of the genetic makeup of the cells from which they were derived, but we can use them then in conjunction with other tools, right? Like we did today for genome editing, CRISPR-Cas9, to generate lots of different models and things like knockouts in which we study the impact of genes on different cellular processes, or knock-ins, right, to assess the impact of maybe reversing a point mutation in a disease state, even reporter cell lines, which we've not talked about today, which have a number of different useful applications as well. We did talk a little bit today about things like electroporation and lipid-based delivery methods that are available for generating these genome-edited iPSCs, cultured, as we've been talking about, under feeder-free conditions, and the importance of different media systems, in our case at Thermo, StemFlex medium for gene editing. We also showed that these iPSC and CRISPR-Cas9 systems are, I think, a really useful tool for assessing things like the influence of different mutations on the functionality of our beloved cells, our beloved iPSCs. In this case, again, today, cardiac cells, right, in the context of dilated cardiomyopathy. Again, I'll share these slides immediately afterwards, but this is some useful tools and resources, especially down at the bottom here. There's loads of content out there, not just on the Thermo website, but on other companies as well, and other biotechnology firms. There's some really, really interesting literature. You could certainly go down big rabbit hole in genome editing, and it'll blow your mind of some of the things that people are doing nowadays. But I wanted you to have this and these tools here in your back pocket moving forward. And I will stop there in the interest of time. But thank you guys very, very much. We hope that the course is going well. We hope that this is interesting for you. We're thrilled to be doing this for our second year in a row. We had good success with it last year. So really appreciate you all being here. And Omar and I are happy to take any questions. Thank you.
Excellent. Thank you, Tom. A couple of things I wanted to add, actually, if you don't mind, Tom.
Please. Yeah.
As you were speaking, I pulled some slides from the CTS deck, Tom, that I think might be interesting to share with
Yeah.
folks, just
Please.
because I wanted to highlight specifically the clonality part about gene editing or the clonal workflow. It's just one slide. If you don't mind, I'm going to share my screen real
Yeah,
quick.
I stopped taking control, so you can. I'll hand the reins to you.
Perfect. Let me see just here real quick. Here's just a couple of slides here, and we can add those to your deck as well, Tom. Can you see the slides okay?
Not yet, no.
Oops. Okay. I might be try this. Oh, I did not even share. Now it should be okay. There we go.
Yep.
So a couple of things I just wanted to mention to folks. So Tom really described very, very well the steps of preparing your cultures and your experiments, and it's very, very important. In fact, if I might add just an anecdotal story, um, right around the time where CRISPR-Cas9 was first taking off around the early 2010s or so, 2013, 2014, something like that. It was so accessible that there's certain folks that were selling them out of their garage. And there was, if you look it up, there was a news story about a person who was selling these kits that he had made in his garage where he was trying to like infect himself or something like that to edit his own cells, which I do not recommend anybody do, right? But these considerations on planning your upstream workflow is very, very important, and I'll mention a little bit more in just a second. But the downstream end, right, is also really important when it comes to iPSCs. If you remember, we talked about how iPSCs don't really like to be singularized, right? So when we're dissociating them or passaging them during routine culture, it's very important to do so in clump format so that they are a little bit more sturdy, right, as you move them from one plate to the next. But during a gene editing experiment, right, you have to actually singularize your cells in order to put them through that workflow. And as Tom mentioned, deliver, your guide, your gene editing tool itself, like a Cas9 or Talon, your donor template potentially, and anything else that might be included there. And to this end, right, when you do the gene editing, the process is very stochastic. And what that means is it really does happen by chance. And I think, Tom, you described this really well of like of the right things, they're being there at the right time. In iPSC biology, if you will, the editing of the genome is accessible because iPSCs tend to have an open conformation, right? So there is access to DNA readily available. But that doesn't mean that every time that you transfect, every single cell is going to be edited, right? So when you do, you end up with what's called a pool, right, of cells. Some cells will have gotten your gene editing tools and would have made the edits that you want, and some will not. And if you go one step further, even the ones that did receive the gene editing tools, where you do notice a change in the genome, just remember that there are two copies of each gene, right? So when you're considering your final desired edit, do you want to be working with a heterozygous a mutant or a homozygous mutant, right? So these are very important considerations during that pool stage. And in order to alleviate this, right, and figure out, okay, what cells am I really working with? Are they a pool of a mixture of non-edited wild type cells as well as mutant cells, or potentially the cells that I want? And so in order to do this, what we do is after the electroporation and the delivery of our gene editing tools, we allow these pools to recover for a little bit and expand them. And then we move to a clonal expansion workflow, right? And this is where you can go and you take your cells that could potentially be different, as you see over here, and you plate single cells in a single well, for example, of a 96 well plate. And the idea is very similar to what we described with reprogramming, right? Is that you want to start with a one cell, knowing what its actual exact genotype is for the locus that you're editing and potentially any non-target effects or off-target effects that may happen. And here, right, because you're plating single cells and single wells, this is where some of the iPSC specific considerations start to come in place, right? What media are you using? What supplements? What matrix becomes very, very important because the emergence of these colonies from single cells will really depend on those factors, right? Aside from that, and again, this is a much newer deck that Tom and I have been working on and didn't make its way to the original training deck, but we can add these slides, Tom. This is just for your reference. Some factors that could be impacting your delivery and your cell engineering workflows when it comes to the sources of the cells and the culture conditions, the processing times, and your electroporation parameters or your delivery parameters, as well as the design of the gene experiment itself, right? And then finally, just to kind of illustrate the point about that clonal expansion, we talked about different PSC medias and different supplements and matrices, right, in the last lectures. And this is an experiment kind of showcasing the importance of these factors during these workflows, right? So if you take those single cells and you plate them in single wells of a 96 well plate after a gene editing experiment, and you do it in what we refer to typically as lean media, right? Essential 8 is the bare minimum that these cells need to maintain pluripotency, right? On a Geltrex or a basal membrane extract matrix, we see around 5% of these cells be able to generate clones that you're able to screen later on, right? And this is quite low, right? This means that if you plate an entire 96 well plate, so you have 96 wells, out of those 96 wells, only 5% are going to be colonies that you can screen, right? So they may not be the ones that you want to work with. These are just colonies that might be the ones that you want to work with. Okay. If we change the supplements and the matrices here, right? Everything else being the same, but we still plate those single cells in essential 8 media, which is rather lean. We add in a ROCK inhibitor to help them attach a little bit better. And we plate them on a laminin-based matrix, which is more supportive for their attachment generally. We see that cloning efficiency rise up to around 15-20%, right? But another big difference we see here is in the media itself, right? So StemFlex medium in Thermo's end was specifically designed for those challenging gene editing workflows. So in this case, we now can see that with a StemFlex medium, a BROC inhibitor, and a laminin substrate, we see up to almost half of that plate become screenable clones that you can work with, which means that your day-to-day is going to be a little bit simpler, easier. You don't have to repeat the same cloning experiment over and over again before you find that one or two clones that are of the right genotype that you want to work with or continue to work with. Sorry, Tom, I did not mean to steal this much, but you know me, I like to talk too much. So just a couple of things there. I'll
Yeah,
stop
super
sharing.
useful, Omar. Thank you.
Of course. And the other thing I also wanted to quickly mention, maybe just a bunch of side stories, but are around the ethics portion. And I'd love to hear what folks are thinking about in the chat. So please free to put whatever you're thinking or come off mute. But this is this is a tool that allows you to practically change the genome of cells, right? So we have to be, or we have to consider the ethical ramifications of the ability to do so, right? And one story I always refer back to, if you haven't heard about it, you should look it up. But there was a researcher who several years ago, I think in 2018, decided to edit a very specific gene called CCR5 that's been associated with immunity from HIV. And he did so on two embryos of two twins. Actually, their names are Lala and Nana, I think. And uh, the issue was the in the design of the experiment, right? Well, there was multiple issues, but proper consent was not taken, and then from patients knowing what is going to happen with their cells and the embryos, but also the design of the experiment was a little bit faulty as well, where the CCR5 gene was targeted, but instead of a very specific amino acid change that is associated with this immunity from HIV, the researcher ended up doing a frame shift that was not associated necessarily with the immunity from HIV with this particular gene. And to even exasperate things, right? When they were doing the editing, they're they had a mixture of edited and non-edited cells. So the embryos were, in that regard, a little mosaic, right? And this caused ripples, honestly, globally, because it was kind of the first instant, like public instance where we realized that, hey, this needs to, we need some guidelines here when it comes to the ethical ramifications. Which brings me to my last point around informed patient consent, right? A lot of you are going to be working with iPSC cells with that you may or may not, edit down the line. But it's very, very important to make sure that anything you're working with when it comes from patient-derived samples, that you have the proper informed consent forms, right? This is a new topic that a lot of people are working with, especially as stem cell therapy is heading towards clinical applications, right? But we have to make sure that we fully inform patients, donors of what these cells are going to be going through, what we intend to use them for, and make sure we adhere to those restrictions, right? This is very, very important. I think that's my last thing. The very, 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 very last thing is last week, Craig Venter, who led the Human Genome Project, passed away. So maybe just a quick shout out, because all of this, honestly, the gene editing, the tremendous work that we're able to do is based off of very foundational work that, Craig and his team really, really pioneered. So a quick shout out to him as well. Just as a last hurrah.
Okay, good. So let me share the quick slide. This one is also very useful information because our center awarded some grant and then collaboration with some official automation team, and then also researchers involved in Johns Hopkins and the Mayo Clinic. And we are developing the automated cell culture system for iPSC reprogramming and the gene editing, especially iPSC reprogramming and the gene editing. For example, if we do base editing or prime editing, there is not much drug selection markers we can use. So in that case, we need to do sub-clone of the something like 100 or 200 up to 300. This one is extremely labor intensive. So therefore we developed the automation system. And this one has a two-legged handler. This one is robotic arm. And then also in the middle is automated confocal microscope. And in the behind, automated 4-degree refrigerator, automated plasticware storage, media storage, automated incubator, which can hold 220 6-well dish. That means that more than 1,000 well and then we can culture together. So that is really saving our researchers' hand. Just to think about if you do the 100 different clone expansion, really a lot of time. So this is the another technological advance for the iPSC reprogramming and the gene editing. And then also nowadays we are incorporating a lot of AI tool. So here, something like about 2 years ago, if we use AI tool, very hard to distinguish good colony versus bad colony. Training the researcher's eye, it takes 6 months. But AI, we trained and then they can immediately determine which colony is better than the other. And then they just, we also developed the algorithm, robotic arm, or automatically pick and then put into the smaller well. And then also when they monitor the compression of the plate, and then they automatically passaging through it. So this is the another technology development. And especially our center, actually recently we are working with some of your local core facilities, something like a mouse core, tissue culture core, organoid core. And then we are providing the core-to-core partnership to provide some of the institution does not have a stem cell core and the gene editing, because this one is definitely can handle more than 100 project at the same time, because this equipment will work without any break, and then 24 hours, even Saturday and the Sunday, even 2 a.m. on Saturday, this machine is continue to work, and then we have a maximum capacity. So therefore, just expect if your institution does not have any stem cell core or gene editing core, or you need to have really a lot of individual colony screening, yes, contact us, and then we will collaborate with your local core facility, and then we will work together. Okay, that is
Looks
the,
great, Kitai.
yeah,
Really nice setup.
yeah,
And I have to I also have just a note to everybody. I really want to emphasize the kind of the importance of learning from people who already have that knowledge base as well, like Kitai in his core, right? These workflows can get very cumbersome very quickly, right? So you don't have to reinvent the wheel, reach out. There's a lot of support that, either, folks like myself and Tom can provide, or the actual work in like Kitai and his teams can actually help you do the projects, right? So use your resources. It's very important.
yeah. Also, just the collaboration with the industrial partner is very important. Without Thermo Fisher, I cannot develop this system. And then also this year, next two lecture, we are talking about bioprinting and AI and the future technology development. Those two special lecture is actually incorporated into this automation system. Without AI, we cannot determine which colony, which well is ready to split, or which colony is ready to pick. That kind of things we need to be incorporate. Bioprinting is also a very important part. For example, if we wanna develop, for example, a steroid of the brain, and then we wanna test the image analysis of some of the brain cell, and then we have an automated microscope. In that case, we can make the bioprinting into 384 well dish. And if we have 10 plate, how many? 3,840 plate. This one is samples. So this N number, can we do with our hand? No. Machine can do it. So that kind of tech, that is the next week's subject of the bioprinting. Following week is about AI incorporation. So what I am saying is that please attend the next week and the following week lecture. And thank you, Omar and Tom, Tom and Omar for excellent lecture. As discussed, we are talking about some Fisher product, but if you look at the underlying principle of the genome engineering strategy, it's very broadly applicable. And then some even other manufacturers' item, if you understand the one product and then what is the principle, you can easily understand the other product. And then you can just compare. This kind of knowledge is always useful. And then also this lecture content will be distributed through the email and then also uploaded to the central YouTube channel after course is complete. And over the next 2 weeks, we will discuss the tissue differentiation from mesoderm. What is the mesodermal tissue? Muscle, bone, blood. Yeah, very important. And then following ectoderm, such as brain, skin, eye, and more. Even if you work on the not directly focusing on this mesodermal or ectodermal tissue, principle of the general tissue differentiation protocol is based on the early embryo development in fetus. So therefore, we are just developing the tissue differentiation protocol. We are chasing what is the normal tissue development in actual fetus. And there's also, if you look at the not only your tissue of the interest, if you are look at the other tissue type, like endoderm and the ectoderm and the mesoderm, you will have very broad knowledge. And then especially this lecture read by Omar and the Tom, and the very short period of time, and then they pick the really core information. So therefore, even if you are on that tissue field, and then please attend it, and then also get the knowledge. And then also next, we are also pleased to have a special speaker, Dr. Glauco Sosa, the expert in bioprinting with a presence in 3D, presents 3D biology to the future. Okay, thanks again for your participation, and we look forward to seeing you next week. Okay.
Thanks, Tom. That was great. Thanks, Kitai.
Thanks, everyone. Have a great week.
Have a good one.
Bye.